So our biological overview today is given by Professor Daniela Krause, who is a specialist in laboratory and transfusion medicine, and she serves as the director of the Institute of Transfusion Medicine at the University of Mainz in Germany. Before moving to Mainz, Daniela was the research group leader at the George Speyer House Institute for Tumor Biology and Experimental Therapy in Frankfurt, and Professor of Cell and Gene Therapy at the Faculty of Medicine of Goethe University. Her research focus is on the role of bone marrow environment in normal hematopoiesis and leukemia-associated hematopoiesis. Professor Krause is co-chair of the annual John Goldman Conference on CML, together with both Professor Cortez and Professor Tim Hughes, and she will share the biological highlights with us now. Thank you, Daniela.
Thank you very much, Nicola, and also thank you to Jorge for starting off on the clinical aspects. So I agree with Jorge that it was a very, very exciting meeting with lots of novel and really innovative insight into the world of CML. I think one can group the biological aspects into several main topics, one of them being the question of resistance, another one being blast crisis, which remains one of the very challenging questions in CML. And then, of course, treatment-free remission and how we can predict it, and which patients basically do well with it and which patients don't. And the other one is sort of immunological aspects of controlling treatment-free remission. And of course, there are some other topics like metabolism and some systems biology approaches. I'll try to give you more specific details now. So there was one talk on resistance, in particular the resistance towards asciminib, so one of the very, very recent developments in the therapeutic arena in CML. And our colleagues from the Hanschel lab in Marburg, Germany, actually showed that there are also other ABL fusions, so not just BCR, but also other fusions to the ABL gene that can cause sensitivity to asciminib. And they were particularly interested, and this was also another group that was working on this, in the role of certain domains within the ABL protein. These are called the SH2 and the SH3 domains that might be causing the sensitivity or the resistance towards asciminib. Another very important topic, and Jorge already alluded to this, is the aspect of other mutations in CML that might be contributing to the outcome. And these mutations are called CHIP, or clonal hematopoiesis of indeterminate potential. And these are mutations that we all acquire as we grow older. And the problem with these mutations is that they can predispose you to other hematological malignancies, such as myelodysplastic syndrome or even frank leukemia. And Jorge already told you that ESH-iCMLf is the most common mutation found also in CML patients, that is associated with a lesser chance to achieve molecular remission after treatment with TKIs. And also there was along those lines, there was a very, very interesting mini symposium on clonal evolution, not just in CML, but also in other cancers, in particular hematological cancers. And you might know that also in myeloma, for example, there is sort of a natural progression of the disease, beginning with monoclonal gammopathy of unknown significance, or MGUS. Then there's clonal evolution towards a smoldering myeloma, and then to the actual plasma cell disease, which is multiple myeloma. So that was one of the diseases that was looked at in this clonal evolution symposium. Another one were clonal diseases in T cells. So for instance, large granulocytic leukemia, LGL leukemias, and others in other types of cells like B cells. We also heard talks about metabolism. There was a very interesting talk from Glasgow, from Vignir Helgeson's group, about the hypoxia that these CML cells are exposed to, in particular in the bone marrow, where there are hypoxic conditions. And this group showed that this hypoxia in the bone marrow niche seems to drive the CML cells to a metabolic switch, making them use carbon as their source, preferentially over other sources for energy. And they also found that certain mitochondrial genes, so the mitochondria are the organelles in the cells that are responsible for the metabolism of a cell, and that these genes and these mitochondria are also completely dysregulated, which probably contribute to resistance to tyrosine kinase inhibitors, etc. They also found, then there was another talk about new combinations of drugs, for instance, tyrosine kinase inhibitor plus ruxalitinib. Ruxalitinib is a inhibitor of JAK2, which you can find mutated in other myeloproliferative neoplasias. And they were looking at that in particular also in relation to the role of the interferon pathway and the combination of interferon with linotinib, which seems to change the expression of certain receptors on the surface of natural killer cells, which are those immune cells that seem to be playing quite an important role in controlling treatment-free remission. Another study also looked at lenalidomide, which is another drug that's been used especially for multiple myeloma, and that seems to also increase the proportion of natural killer cells and T-cell proliferation and activation. Now, looking at sort of the genomic studies that were shown at the conference, it was very interesting to see that the concept of the leukemic stem cells is not quite as straightforward as we always thought. It seems actually as if these leukemic stem cells are quite heterogeneous. There seem to be some that are primed towards a certain lineage. As you know, CML blast crisis can be of the myeloid or the lymphoid lineage. And it seems as if a patient might have leukemic stem cells that are more primed towards either the myeloid or the lymphoid lineage. And that there even seem to be leukemic stem cells that are sort of particular to the relapse state of a CML, for example, versus remission leukemic stem cells. And these remission leukemic stem cells have a signature that very closely resembles, or that has aspects of the interferon pathway. As you know, back in the old days, patients with CML were treated with interferon. And so it seems as if interferon is really quite an important pathway in these leukemic stem cells. We know that this lineage bias in these leukemic stem cells sensitizes to certain tyrosine kinase inhibitors, and it seems in particular the red cell progenitors are quite sensitive to tyrosine kinase inhibitors. And also the epigenetic state of a CML stem cell also seems to contribute to sensitivity. There was another interesting talk by Dr. Carlson from Lund in Sweden, who reported on the expression of CD35 as another very important leukemic stem cell marker in CML. It seems to epigenetically define a hematopoietic stem cell or leukemic stem cell, as it seems to describe a cell that has self-renewal potential, and it sort of captures the enhancer signature, the epigenetic enhancer signature, and stem cell activity. We had a very important and interesting keynote address by Dr. Bonnet from London at the Crick Institute. Dr. Bonnet talked about the benefits of the bone marrow microenvironment and the benefits of understanding the bone marrow microenvironment towards CML understanding and acute myeloid leukemia understanding. In particular, Dr. Bonnet and others have worked on the humanized bone marrow microenvironment. For instance, they create polymers or little scaffolds that they can implant under the skin of mice and look at how certain hematological malignancies engraft, and then obviously you can study them quite well in immunodeficient mice. She was also looking at the myelodysplastic syndrome in her research. Another keynote addresses looked at the immunology of treatment failure. As mentioned, there are these natural killer cells that seem to be responsible for controlling treatment-free remission, and they seem to be less adaptive in CML and less functional, which might contribute to why some patients relapse after stopping treatment. There are also myeloid-derived suppressor cells, another immune cell type that might be increased in CML and thereby basically suppressing the immune system and allowing the disease to progress. It is clear that these adaptive natural killer cells seem to be predictive of a good outcome after stopping treatment. And also CLPs, or common lymphoid progenitors, so basically a progenitor cell of the B cells and the T cells, can predict relapse. And also a certain signaling pathway called mTORC1 seems to be quite predictive of relapse or not. As mentioned, there were quite a few talks on blast crisis, and it seems as if there are, as one can imagine, quite profound differences between chronic phase and blast crisis CML cells. The blast crisis cells seem to have quite striking differences with regards to metabolic pathways. So the OXPHOS pathway is highly upregulated in the blast crisis cells. They have more protein processing. There is more signaling via interleukin 12. And it seems that a general inflammatory signature, in particular meaning interleukin 6, JAK-STAT pathway, and interleukin 2 and STAT5 seem to be more active in these blast crisis cells. Then other colleagues were looking at certain, so in B-cerebral positive acute lymphoblastic leukemia, our colleagues were looking at certain clusters that might be predictive of outcome depending on certain gene expression. And so this was Deborah White from Adelaide. She was looking at the association of certain genes such as CDKN2AB or Icaros for the progression or the development of B-cerebral positive acute lymphoblastic leukemia. So with other words, seems to be a particular set of genes in addition to B-cerebral that seem to predispose a patient to blast crisis. And also the question of how long you've had your CML seems to also be a very important factor in which patient might progress to blast crisis. So the biomarkers basically of blast crisis seem to be stemness, inflammation, and quiescence. Thus, that was really quite novel. We had, oh, and let me just tell you, there was one other very interesting abstract given by Dr. Van Etten from Irvine in California. He presented basically a systems biology approach in which you can predict the response and the resistance to TKI treatment. And this was really a mathematical model with some mouse work that might predict which patient under what constellation and under what mathematical constellation of leukemic stem cells versus normal stem cells might progress. Actually interleukin 3 and another cytokine, CCL3, were implicated in this process. We had two very exciting workshops. This time, these workshops are great because either they sort of present an exciting technology, or they present sort of new concepts and new developments, new therapies. The technology-based workshop was on a technology called spatial transcriptomics. And the beauty of this technology is that you can take, for instance, a bone marrow specimen, and you don't just do what we call RNA sequencing, but it's actually, you can correlate the gene signature of these cells inside the bone marrow with their location. So let's say all the cells that are at the tip of the bone have a certain signature compared to those at the end of the bone, to make it simple. And there we had a very interesting talk actually about the B cells in lupus, so an autoimmune disease, versus those in myeloma. And so the gene signatures in these different locations in the bone marrow were compared, and they found certain niches enriched in myeloma cells in the bone marrow, and there seemed to be very immunosuppressive environments within this bone marrow. And the second workshop was on cell therapy. So as you might know, in 2012, there was a major breakthrough where a young girl in Philadelphia was treated with these immunologically active T cells. And these T cells had been manipulated to really target the leukemia cells. This is a treatment very successful in acute lymphoblastic leukemia and chronic lymphocytic leukemia. And it's now being spread also to other, to solid tumors, and to once trying also in AML, and increasingly also in autoimmune disorders, for example. So there was one study presented from Boston by Marcela Maus about a new targeted therapy in the form of cells. So basically these are called CAR T cells, chimeric antigen receptor T cells, against a new antigen, which was CD37. And she showed the data of this trial that they had done on 5 patients who responded quite well. But the problem with these treatments is that many patients have a severe cytokine release syndrome. So the sudden attack by immune cells can really release cytokines, which can make the patient very, very sick. The second talk in this workshop was about these natural killer cells, which I was talking about, which can control treatment-free remission, but very interestingly, you can also manipulate those and use them therapeutically. So the benefit here is that usually you don't have the cytokine release syndrome. There's no problem with graft versus host disease. And this colleague, Dr. Romay from Boston, he was working with cytokine-induced memory-like natural killer cells. And he was able to show that he could really extend the half-life of these NK cells in the patient, which has been a huge problem with this NK cell therapy, namely that the NK cells just don't last very long. So that was a great progress. And he was now even working on the microbiota. So these are all the bacteria you have in your gut, which he's also trying to manipulate in order to provide a better platform for these cellular therapies. And then the last talk was also about the microbiome, which is really an up-and-coming field because the bacteria in your gut can predict outcome to treatments in CML and also in other malignancies. For instance, if a patient has a hematopoietic stem cell transplant and gets lots of conditioning therapy in the way of chemotherapy and radiation, that significantly changes the composition of the bacteria in a patient's gut. And that itself has profound influence on the outcome after these therapies. So there are certain types of bacteria, for instance, the Enterococcus. If you have very dominant Enterococcus, for example, the patient may be more prone to graft versus host disease or mortality. Then he was studying the effects of changing to a different diet, a lactose-free diet, which might reduce graft versus host disease. And there are even, attempts to do fecal transplants in order to make a patient benefit after these really important treatments like stem cell therapy. And it seems that the Clostridia, another type of bacteria, seem to be very beneficial. And it also seems to be beneficial to have a very diverse microbiome before stem cell transplantation. So with other words, the microbiome is much more important than we've thought for a long time. We know it also plays a role in cardiovascular disease, for example. So I think those are sort of the highlights of the conference. And I think that is it from my perspective, and I'm happy to take your questions.
Thank you so much, Daniela. Yes, again, so many highlights and so many new advances coming through. It's quite amazing. Every year we see the same thing. So yes, we are now open for questions from the audience. Daniela, I'll give you a break. There's been a couple come through on the chat, so maybe we'll start with those. So again, for those of you in the audience, if you'd like to come and ask your questions directly to our speakers, you are more than welcome. But to start with, I will read Ekaterina Cheseve's question. So Jorge, what is your opinion of how to treat a T315I negative CML chronic phase patient resistant to asciminib in third line or more if BMT is not available, age or comorbidities? Is there any reason of dose increasing, and what combination strategies may be discussed?
Well, this question raises an important issue, which is what's the right dose of asciminib on a patient without a T359 mutation. The standard dose is 40 milligrams twice daily or 80 milligrams once a day. But as we know, asciminib is quite safe all the way to 200 milligrams twice daily, which is the dose that we use in T359. So is there room for higher doses in some settings? We know that there are other mutations that, at least in preclinical work, in vitro, require higher concentrations of asciminib, like the F359 and a few others. So I think that certainly if there are any of those mutations, I would consider higher doses. There is actually a study going on right now, which is called the Ask to Escalate 2, which is exploring that, what on patients who start with standard dose who don't respond, if we escalate the dose, can we get better responses? So we don't have prospective data on that yet, but I think that in selected patients that could be considered. Now, of course, you also have ponatinib, and ponatinib is a good drug, and we should consider it for patients where we've tried asciminib and they're not responding. It has good efficacy in the same patient population, patients with 2 or more TKIs, patients with T359. So that is another alternative. So what is better? We don't know. We haven't done the studies, but both would be reasonable options.
Okay, thank you. And I guess sort of another one for you, Jorge, around the question of ruxalitinib. So the question is, what is the criteria for ruxalitinib, JAK2 positive and or other mutations? I think that's referring to the study of Professor Sweet.
No, the principle of this study was not that there is a JAK2 mutation. It's just the fact that the JAK2 and STAT1 activation is one of the pathways that maintain these early progenitors alive, even in the presence of a tyrosine kinase inhibitor. So it's not through mutations. It's just a completely different concept than what we see in myeloproliferative neoplasms. Of course, we do see an occasional patient with CML that has a mutation with JAK2, but in this study, they were not selected by JAK2 mutations. It's just the consideration that this JAK-STAT pathway is activated. It maintains. It causes that persistence of these early progenitors. So there's no need to. Now, of course, this is not an approved indication for ruxalitinib, but if it were, there would be no need to check for mutations for these purposes. It would be just the principle of the activation of the pathway.
Thank you. Daniela, perhaps a question for you. So we see many new TKIs on the pipeline, but where do we stand when talking about finding an actual definitive cure for CML? So what is the hottest target for today?
Yes, I think Jorge can also answer that. I think one of the important question is, I think, what patients can we take off of the TKI, and what probably does the immune system do to this? So I strongly believe in the combination of therapies. So probably, a TKI plus immunologically active drug, possibly plus something else. That is sort of my opinion. I think there is no just one hot target right now. We're following many leads. And I think, in my personal opinion, this is a personal opinion, it will be a combination of several aspects. But I'll let Jorge answer this one as well.
I fully agree. I think that we probably, the patients that we can get with the TKI, we're probably maxed out. We're probably going to get to the maximum with a drug like asciminib. It's more potent, and we're going to get to the highest rate of MR4.5 sustained and so on. But it's still going to hit a ceiling of only certain patients, and we know that even those patients, they relapse. Many of them do. Probably about half of them relapse once we stop therapy. So there has to come from something else. And I also agree with Daniela that combinations, but I think that the combination strategy is going to have to depend on the patient. We're going to have to figure out a way to understand what is more significant for a given patient. If we look at this Ruxalitinib study, for example, the difference is very significant, but it's still only a fraction of patients who reach that MR4.5. So certainly for some patients, that pathway became important. It is important, and blocking it works. But for many patients, it doesn't. So what else is there? I am a strong believer also in the immunology approach, but what immunology approach? What do we need to do for that? So I think we need to still figure out what works for what patient and how we can identify those different approaches in different patients.
Yep, fair enough. A specific question that's come through that may or may not be able to answer. So with the Blast Crisis study with ponatinib plus AZA, did it include transplant eligible patients? And if so, how many were transplanted?
Yes, that's a good question. Yes, they were included. And actually, a good number of patients were transplanted. Remember, this was a small study, but 7 of the 19 patients were able to go to a transplant. Evidently, that's what we would like to do with these patients, get them to a good response, as good as possible, and get them to a transplant. But they also showed that some patients that did not receive a transplant, for whatever reason, we know that not everybody has a donor, not everybody's eligible, and so on. Many of these patients who did not get a transplant still were in a sustained remission, at least at the time of the last follow-up. So yes, I think that the ideal would be some combination, something like this, followed by a transplant. But it is good to have strategies that may provide durable remissions, even in the absence of transplant, for those who cannot get a transplant.
Thank you. Any further questions from the audience? So if not, I might sort of, we are over time, so I might just conclude by asking you both a final question. Really, so Daniela, you outlined many highlights and many areas of research. Could you sort of outline one particular that you think is the most promising area of research coming forward, with perhaps particular regard to clinical need?
Yes, I think, and yes, I mentioned several, and I do believe in all these different approaches, but I do think the immunological aspects, in particular in CML, are going to be key, and understanding those, and as Jorge said, understanding in each individual patient what the immunological factors are will be key. CML is the most immune sensitive disease, and that's how basically transplantation started on CML patients. And I believe that there's something to be gained from that, and the fact that we all have BCR-ABL in us, but we can control it, but some people cannot and develop the disease. That really, it fascinates me, and I believe that there's a lot more we need to do there, and that's what I would place my money on if I had to.
Thank you. And Jorge, so a similar question for you. So what particularly stood out for you in the biology sessions that you thought would be coming through for clinical use in the near future?
I concur with Daniela that I believe that we only played a little bit with the immunology question. We've gotten too excited with the very good efficacy of these tyrosine kinase inhibitors, and we should. I mean, they're great drugs and they've worked and they've helped many patients, but we still have many gaps that we have not been. I think we're probably going to reach a limit of what they can do, and for many settings, including the TFR, but also others like what do we do with patients that have ASXL1 mutation and things like that. We need to explore these combinations. I would put my money on the immunology aspect of it, but what, how? Is it a checkpoint inhibitor? Is it what type of immunology? There's been interferon with some immunologic aspects of interferon, but we don't know yet what. So to me, we need to understand better what elements of immunologic stimulation or manipulation can help patients and can help us overcome these areas where we've reached a maximum. The other thing is that we are starting to uncover these and understand it better, but we still have a lot of work to do on a disease that we've always felt that it was such a homogeneous disease. Everybody has BCR-ABL, and it's that simple. You have BCR-ABL, you get a TKI, gone. We've understood now that it's such a molecularly complex disease, and that complexity is giving us a lot of different angles and different questions that we still need to try to answer. So I think that's what we're going to be seeing for the next several years in our meeting and in others.
Thank you. Thank you both. Again, really appreciate the time that you go to, having come away from the meeting so recently, to take the time again to give us the highlights of the conference. And it'll be really interesting, sort of given your perspectives, where we actually go to for the meeting next year, which will once again be in Portugal. Now, Jorge, I don't know whether you have the dates for people's diary handy.
Yes, we do have the dates, although I don't have them right in front of me. It's in October this year. I believe it's October 12th, 13th, something like that. But we'll post them in the ESH-iCMLf webpage shortly.
We certainly will. And for those of you who haven't signed up for the ESH-iCMLf news, you can do so on our website, where you can also find lots of other news and clinical case discussions, papers of the month, and all the news on our programs and activities. But that does conclude our clinical conversation on the John Goldman Conference highlights. And again, please join me in thanking Professor Krause and Professor Cortes for their time. We hope to see you at the highlights of ASH meeting, which will be in January. And in the meantime, if you do have any specific questions, please don't hesitate to reach out to the foundation by email or via social media.